The big question that we have been focused on for several decades now is what kinds of changes in electrical activity underlie both the learning, the formation of memory, and the sustained expression of memory. Our lab is located at Indiana University Bloomington, and the creature that we work with is called Hermesenda crassicornis. It's a very brightly colored nudibranch mollusk that's found all along the Pacific coast of North America. And this nervous system is a relatively simple one. Total of about 20,000 neurons. That compares to the 100 billion neurons or so given as estimates for the human brain. And it was the work in our initial decade or so that basically focused us on specific classes of ion channels, both voltage-gated channels and calcium-activated channels. And so we used selective pharmacology to basically show that we could block each different class of voltage and calcium-activated potassium channels, and that when we did so, that partially mimicked the effects of behavioral conditioning.
Our lab has been really interested in voltage-gated potassium channels, specifically the Kv1 family members. And we're really interested in them as potential early targets for Alzheimer's disease. So we're looking at ways that they may be suppressed, especially under exposure to amyloid beta. So when our cells are exposed to amyloid beta, the Kv1.1 and 1.2 channels, or the heterotetramers, they lose 50% of their current within 30 minutes. So we see a really rapid and robust suppression. When these channels are suppressed, they lead to hyperexcitability and excess glutamate release. And this could in turn also lead to synaptotoxicity and neurotoxicity. And this could also relate to early Alzheimer's disease progression through early loss of synapses that we see before cognitive deficits. What's been most interesting is that throughout the project, we have been able to test a number of different compounds. And actually, one of them, we found that when we incubated our oocytes with this compound, it completely prevented suppression by amyloid beta for our ion channels, so Kv1.1 and 1.2. And that compound was a broad-spectrum tyrosine kinase inhibitor. So our current next steps are really exploring the calcium dependency and especially the tyrosine kinase dependency. So we're looking at a specific tyrosine kinase of interest, and potentially we'll look at more.
We work with xenopus oocytes that we inject with cRNA encoding for the potassium channels that we're trying to study, as well as PIC2, which is the protein tyrosine kinase. So then we add amyloid beta to the solution and we model that suppression and collect data on that.
I work with amyloid beta potassium channels 1.1 and 1.2. We usually look at some mutants that DNA selects, and sometimes we inject substances such as like PMA or amyloid beta and measure the membrane of those cells and see how they kind of like deteriorate over time when being expressed in those solutions.
During my first semester as a freshman, I came across Dr. Farley's research, and it just really compelled me because it was something that I hadn't ever studied on my own, and this lab has really brought on a strong foundation, understanding the biological mechanisms behind those diseases.
We have known for a long time that when people become addicted to nicotine, it stimulates, it activates, and then desensitizes many different molecularly distinct neuronal nicotinic acetylcholine receptors. The second most abundant high-affinity nicotine binding site within the brain is the so-called alpha-7 receptor. And we spent a great deal of time basically identifying which are the calcium-dependent signaling pathways that are ultimately responsible for the upregulation of the alpha-7 receptor. And so we have basically started to screen FDA-approved libraries of chemical compounds. These are primarily drugs that were developed for other reasons. And what we want to know is, are any of these compounds capable of acting as what's called a positive allosteric modulator, or a PAM, at the alpha-7 receptor? And we have, in fact, identified a number of hits, and we've shared that information with our partners at Rose Research, and they are in the process of testing some of the more promising compounds with their smoking cessation protocols to see if they work. And we think that this is the correct strategy to pursue.